I'm sorry, the speaker is muted. Please unmute. Thank you.
Sorry about that. No problem. You good? Thank you. All right. Okay. Yeah. So the second challenge is that we observe like often reads in the intron regions, and that could be due to different reasons. The first one is incomplete splicing. So splicing is a dynamic process that cuts the intron out from the pre-mRNA to get the mature mRNA. And RNA-seq is a snapshot. So it could be that in the middle of the splicing, a partially spliced RNA was captured and sequenced. And those reads from the intron regions could be also a really functional protein with the intron retentions. And the reads could also be from intronic RNAs or even DNA contamination. Third, to accurately assemble a full-length transcript, we have to identify the splicing, we have to identify the starting and ending sites, and also the internal junctions. But the signal for the starting and ending sites are often quite weak because they are easily mixed with the either intronic or intergenic reads. And last but not least, some exons are tiny, like only a few base pairs, which makes it very challenging to be correctly aligned to the reference genome. And also, we have homologous genes, and therefore many reads are multiple aligned. So all those factors make the transcript assembly a very challenging computational problem. I cannot switch slide. Oh, now it works. You might have heard that like long reads can produce full-length molecules. So one long read, one molecule. But this is only partially true because a large portion of long reads actually are truncated. So they are fragments, they are not full-length. So therefore, assembly is still needed for long reads. And currently, the long read sequencing reads still, I would say, have quite high error rate. And therefore, we will see this kind of dented alignment in the junctions. Like this is a real example. You can see it's quite challenging to accurately identify the splicing position. And 2 years ago, the community conducted a research called the long-reads RNA-seq Genome Assembly and Assessment Project, or LR-GASP. The paper was out last year, and this work extensively benchmarked the existing assembly methods. And the conclusion is that existing methods are quite far from being satisfying in terms of assembly accuracy. All right, so now let's see how to do assembly. So my work focused on reference-based assembly. So the first step is to align all the RNA-seq reads to the reference genome. And this is a example showing the alignment in the gene locus. So the real situation often is more complicated. So some RNA-seq reads will be aligned like in one piece, but some reads will be splitly aligned, like this example, because they contain a junction. And we will use those like splicing junctions to cut the reference genome into different segments, like A, B, C, D, E, and F. And each segment will become a node in this called splice graph. So junctions will be modeled as edges in this graph, and the edge weight will be calculated as the number of reads that supporting a junction. Like here, we have 2 reads supporting this junction from vertex A to D, so the edge weight is 2. And we also add a source vertex S and sink a vertex T to the splice graph. So as you can see, like nodes corresponds to exons or partial exons. And therefore, transcripts corresponds to the paths in the splice graph from S to T, and we call them ST paths. Naturally, we formulate assembly in this way. So the input is a splice graph, and the output is a bunch of weighted ST paths. So each ST path will predict a transcript, and its weight predicts the expression abundance. So of course, what is good set of ST paths? Or in other words, what are the objectives to optimize, right? And also how to find those ST paths. In other words, what is the algorithm? So different assembly methods answer those 2 questions differently. So we approached the transcript assembly first starting from a theoretical perspective. We asked, if the data is perfect, can we do assembly well? By perfect data, we mean, so the uniform from a molecule is, the coverage from a molecule is uniform, and all the reads can be perfectly aligned to the reference genome. So if we make this strong assumption, then the splice graph becomes a flow graph. And we started this formulation. So given a flow graph, we want to find a set of weighted ST paths from the flow graph so that the flow can be perfectly fit, and that the number of ST paths is minimized. So the minimization of ST paths follows from the parsimony assumption. So we started this formulation, and we get some interesting theoretical results. So we give a formula to characterize the optimality gap. The gap is defined as the known upper bound and the number of ST paths in the optimal solution. And based on this theoretical result, we designed an efficient heuristic. And the heuristic works very well on perfect data, like on flow graphs. We're happy to see that this theoretical work gets a lot of attention from the community. So here I listed a bunch of follow-up papers. So as I said, this heuristic for flow decomposition works very well on perfect data. But when we apply them to the real data, they don't work well. So this motivated us to design a practical assembler, and we did it, called Scallop. Was published in Nature Biotechnology a few years ago. So one lesson we learned from the theoretical study is that the splice graph misses very important information in the alignment. So the information is the reads that span multiple vertices. Like those two orange rays, they span vertices B, C, and D, and this blue one spans vertex A, D, and F. So such reads are not rare because many exons are quite short, like less than 50 base pairs. So therefore, even short reads RNA-seq data may have many reads that span more than two vertices. And those reads cannot be directly modeled by the graph. So a graph can only build connection between two vertices. So we extract those reads that span more than two vertices and model them as paths in the sparse graph. So quite naturally, right? Like those two orange reads can be represented as a phasing path called like B, C, and D. So now we have an improved formulation. So the input is a sparse graph and a bunch of phasing paths. And the output is the same, a bunch of weighted ST paths. And we designed three objectives. So the first one, and also the most important one, is to preserve all the phasing paths, or phase preserving. So it means like each phasing path must appear in at least one ST paths. Again, this is quite natural because like phasing paths is representing like single read, right? So a read must come from some transcript. So the second objective is to use smaller number of ST paths as possible, again, following the parsimony assumption. The third one is that we hope those ST paths collectively fit the given sparse graph as much as we can. And this is equivalent to maximizing likelihood. So we designed heuristic for this formulation. So the algorithm will iteratively decompose individual vertices to simplify the sparse graph, so on and so forth, until it is fully decomposed, until it becomes a bunch of ST paths. So in this toy example, you can see we first decompose B, and then A, and then C. Now it becomes a set of paths, right? S, B, C, T, and so on. So remember, we have three objectives to optimize. That is a really ambitious goal. So we can only guarantee the first one, and we will guarantee the first one. That is like to preserve all the phasing paths by not breaking any of them during this like decomposition process. And for the second and the third one, we cannot guarantee that, but we will try to optimize them as well. So let's see how we decompose individual vertices and how we take care of those objectives. So if we can split a node into two while keeping on the phasing paths, we do it. We call it splittable vertex. And the task is to split this one into two and reassign all the adjacent edges so that each of the new vertices is as balanced as possible. And this task can be formulated as a subset sum problem and efficiently solved by a dynamic programming algorithm. So now let's check the objectives. So again, so in the reassigning process, we will not break any of the phasing paths. So red phasing path still here, blue one is still here, green one is still here. So we preserve all the phasing paths. And you can see after decomposition, we have one more node, but the number of edges don't change. So in fact, this will reduce the upper bound according to the theoretical work I just mentioned. So this will help to use a smaller number of ST paths. And the last, we will try to make each of the new nodes as balanced as possible. And this will help with the objective number 3 to fit the given sparse graph. So if a node is not splittable, then it's unsplittable. And we will use a different procedure to decompose that. So in this case, we will fully decompose a node just following all the phasing paths. And we will have a bunch of new edges corresponding to the phasing paths. And we will calculate the weight of those edges using a linear programming to directly fit the width of the existing edges. Again, we can check the objectives. Again, the phasing paths will be preserved, and we directly optimize the objective number 3. So we can iteratively apply those two procedures, and eventually the graph will be fully decomposed. And this concludes the methods. So let's see some results. So we compare our assembler scale-up with two, like back then, leading assemblers, strength high and transcom. And we compare those methods on 5 or 6 samples corresponding to the 5 figures here. So each method will predict a bunch of transcripts. And we use this ensemble annotation as ground truth. So we will come to this later. So if a predicted transcript matches the intron chain of one transcript in the ensemble annotation, we think it's known or correct. So with that, we can calculate precision and recall. And by changing the abundance of the predicted transcripts, we will get a precision recall curve. And you can see the curve for scallop was above the curves for the other two methods, indicating that scallops gives a higher assembly accuracy. So let's move on to the next tool called Scallop LR. It's a assembler for PacBio long reads. And this project was led by Laura, a PhD student in Carl's group. First of all, the core idea of scallop, like phase preserving, is suitable for long reads because long reads can be modeled as long phasing paths in the graph, so that the phase preserving decomposition will make use of that. And we designed two tailored methods for PacBio reads. So PacBio reads will label each read whether it contains a 5' or 3'. This is very useful information that can be used to accurately identify the transcript's starting and ending size. And we use that to improve the quality of the sparse graph. And also we added a post-decomposition clustering procedure to classify those transcripts that only differ a few base pairs as one transcript. This is to address the higher error rates in the PacBio reads. Again, let's see some results. We compared scallop LR with two other methods for long reads, strength high and RNA-seq. So each predicted transcript will be put into one of those 4 categories. So if your predicted one matches a annotated one with identity of 95 or above, that predicted transcript will be put in this like highly accurate category, and so on for 75 to 95, 50 to 75%, and below 50. So as you can see, scallop LR can predict many more highly confident transcripts than the other two tools. All right, let's move on to the next one called Scallop 2, led by 2 PhD students in my group, Qimin and Qian. So Scallop 2 is a examiner for multi-end RNA-seq data. So what is multi-end RNA-seq data? So probably you've heard of paired end RNA-seq data. So for example, in a typical Illumina sequencing process, so those molecules like mRNAs will be fragmented. So the 2 ends of a fragment will be sequenced, leading to like paired end reads. Like A1 and A2 are the 2 ends of a paired end read. So of course, like the 2 ends A1 and A2 come from the same molecule, right? Because they come from the same fragment. So barcode techniques like UMI is widely used in recent sequencing techniques. Here I use SMART-seq3, a single-cell RNA-seq protocol as the example. So the same UMI label the copies of the same molecule. So again, like in this protocol, all those molecules will be fragmented and sequenced. And if we see the same UMI from 2 ends, like let's see here, A1 and C1, then we know that they come from the same molecule. And therefore, all those 4 ends, A1, A2, C1, C2, are known to come from the same molecule. And we name them like multi-end reads. In short, like those ends in one multi-end reads come from the same molecule. And paired end reads can be regarded as a special case of multi-end reads. So multi-end reads can be naturally modeled as multi-end phasing paths in the context of a splice graph. So like in this example, those four ends are mapped to node A, C, and D, and F respectively. Then very naturally, this multi-end read can be represented as a multi-end phasing path. Actually, it consists of four paths in the graph. So the assembly of multi-end data can be formulated in this way. Again, the input is splice graph and a bunch of multi-end phasing paths. And the output would be the same, ST paths and their expression boundness. And we can still reuse the three objectives in Scallop, like first, preserve of multi-end phasing paths because we want to make better use of them, and also use a smaller number of ST paths to follow the parsimony principle. And also we want that those paths collectively explain the splice graph very well. So this is a framework of Scallop 2 to solve the previous formulation. Given the splice graph and multi-end phasing paths, we first do bridging. Bridging is to transform those multi-end phasing paths into like single-end phasing path by closing the gaps between consecutive ends. So we observe that. So Reed's filter bridge actually is quite helpful. Actually, is informative because they indicate that the splice graph is incorrect, and we can use those reads to refine the splice graph. After that, we will decompose the refined graph to get those transcripts. So step one, bridging. So let's see this example with three ends, A, C, D, and F. So bridging is sort of amounts to find a path from A to C and find a path from D to F. But the question is, there might be multiple paths from A to C and multiple paths from D to F. So which one to pick? So we formulate this problem in this way. We want to find a path so that the bottleneck is maximized. In other words, the smallest edge on the path is maximized. And that formulation can be solved efficiently by a dynamic programming algorithm. So after bridging, this like multi-end phasing path becomes a simple path, right? Or single-end phasing path. And that types of single-end phasing paths can be handled by the phase-preserving graph decomposition. So step two, refining. So again, let's see this example, a multi-end read with two ends, but there's no path from B to E. And we observe that this is often due to missing junctions in the data. So leading to this incorrect ending vertex C and starting vertex D. So we designed heuristic to determine those like false starting and ending vertices. So we first connect like consecutive ending and starting vertices and add a small weight. And then we will do bridging on this like augmented splice graph. And after that, we will check how many times this like pseudo edge was used. And if this edge was used like a significant number of times, we know that those two nodes are false nodes, and we will remove them from the splice graph. By doing that, the quality of the splice graphs will be improved, which is important in improving the assembly accuracy. So last, we designed an improved decomposition algorithm. So remember in Scallop, we will decompose unsplittable vertices like splittable vertices and so on. But what if there are like many unsplittable vertices? So which one to pick to decompose first, following which order? So we realized that the order actually matters. So in Scallop 2, we designed a new consensus voting approach. So we will decompose the graph n times. And each time we will use a random order to split the vertices in a category. And doing that, will give us a set n sets of ST paths, and we will keep them. We will keep only those that appear in n/2 sets. All right. Again, this concludes the methods part. Let's see some results. So we compared scallop 2 with strain type 2 scallop and class 2 assemblers. And on data set, it's a single cell data set from SmartSeq3 protocol with 192 cells. So the x-axis gives the precision and the y-axis giving the matching transcripts to a annotation, which is proportional to recall. And each point corresponds to a cell. And you can see the accuracies of the scallop 2 assemblies are higher than the assemblies of other methods. We also compared those methods Illumina paired end data on 6 RNA-seq samples. So those points, again, corresponds to the accuracy. And we draw the precision recall curve of scallop 2 by changing the predicted abundance. And again, you can see the curve is above the points of other methods, again, showing that scallop 2 gives higher accuracy. All right, next one, Terrace. So this is a tool for assembly circular RNAs and led by Tasfia, Chen, and Carl in my group. So why circular RNAs? So circular RNAs are widely expressed in tissues and play important roles. So the study of function of circular RNAs is quite active research area currently. And circular RNAs, they don't have 3' end, they don't have 5' end, and therefore they are much stable than linear RNAs. So for example, like their is typically 2.5 times longer than linear RNAs. And also because they are very stable, they are reliable disease biomarkers and therapeutic targets. Many methods have been proposed to study circular RNAs from RNA-seq data, but most of them focus on the identification of back-splicing junctions, which is important, but they cannot be used to assemble the full-length circular RNAs. And the full-length assembler is our focus. And among those that full-length assemblers, they often require a reference annotation. Therefore, they cannot be used for like non-model species, which we don't have a reference annotation. And our goal is to develop a full-length assembler that don't necessarily depend on the reference annotation. And also the accuracy of existing methods are not high, and we hope to improve the assembly accuracy. So this is the framework of Terrace. Again, we start with RNA-seq alignments. The first step is to identify reads with back-splicing junctions. And then in the second step, we will assemble each read with back-splicing junction into a circular RNA. And the same circular RNA will be then merged. And in the third step, we will train a machine learning model to assign a confidence score for each assembled circular RNA, which will be helpful to users. So we use two sources of information to identify reads with back-splicing junctions. So the first one is from chimeric alignments. So this is an example. This is one end of length 90 base pairs. So the right 40 base pairs was aligned here, but the left 50 base pairs was aligned there. So this is a strong signal indicating there's back-splicing in this end. And we collect those reads. We also designed a new mapping method to identify back-splicing junctions. Again, we start with reads that are partially aligned, like here. So the left 30 base pairs are not aligned, and we will try to align them into possible back-splicing positions. And if the alignment score is above threshold, we also collect them. So in step 2, we will assemble each read with back-splicing junction into a full-length circular RNA. So we assume we have the paired end data. So this is the first end, like R1, with back-splicing junction, and we assume the second end, R2, is here. So you can see like assembling full-length circular RNA is equivalent to closing the gaps, right? So this is exactly bridging. So we already know how to do bridging. So we reuse the bridging procedure in Scallop 2 here. So it's nice to see a module we developed previously can also be used here. So in practice, we not just generate one path, we actually generate multiple paths. And if the reference annotation is available, we also generate more paths from the annotation. And then we select one following some selection heuristics. So now we have a bunch of assembled transcripts. We want to assign each one of them a confidence score. To do that, we extract 13 features. For example, the soft clip length, the length of the unmapped portion. So the longer actually, the more reliable the backsplicing junction will be. Also, we use how many reads supporting this circular RNA as features. And also, remember in bridging, we will calculate the bottleneck weight, and that is also an informative feature. And using those features, we train a random forest model, which will assign a confidence score for each circular RNA. So now let's see some results. So first, we compare methods without annotation, without a reference transcriptome. So without a reference annotation, 34 is the only method we identify. So therefore, we compared Teros with it on 5 like tissues corresponding to the 5 figures here. So this point, the purple one gives the accuracy of Teros, and this red one gives the accuracy of 34, and the number next to it gives the F1 score. You can see the F1 score of Teros is higher than that of 34 over all those tissues. And we also draw 2 precision recall curves. One is by changing the random forest score. The other is by changing the predicted abundance. And you can see the random forest score corresponding to the green line here is above the purple line, which shows that the random forest model gives better balance of precision and recall. So this is the result when the reference annotation is available. So we are able to identify more methods to compare with. And the conclusion is the same. We are more accurate, and the random forest method gives a higher, gives a better precision recall curve. All right, so I hope you are not bored. This tool is called ALACH. It's to assemble multiple RNA-seq samples, and it was led by my students, Chen and Qiming. So this problem is known as meta-assembly. So the input is a set of samples or set of cells instead of individual sample cell. So the output is the union of the expressed transcripts from all the samples or cells. So to do meta-assembly, of course, we can assemble individual sample or cell separately and independently, and then union the outcomes. But this approach is not desirable because it ignores the shared information across samples. We can also do meta-assembly in another way, like we first pull all the reads together as a single sample and then assemble it. But this approach does not work well either because it ignores the information of individual samples. And also, it will significantly increase the complexity of assembly. So assembly from single sample is already quite complicated. So putting all the reads together is not a good idea. So we developed this method called ALACH, and this is framework of ALACH. So we start with the realignment of individual sample. We first build splice graph for individual samples, and we will build a combined graph for the graphs from the same gene locus. And then we do bridging to bridge paired end reads. And for those reads that are not bridged in individual sample, we will use the combined graph to bridge in the hope to borrow junctions or exons from other samples. So after bridging, so those phasing paths will be used to refine those graphs. Next, we will decompose all those graphs, like individual graphs and combined graphs, to give us a bunch of candidate transcripts. And we will then train a random forest model to score each candidate transcript. So so the same transcripts might be assembled from different graphs, and in that case, we will merge them into one, and their score will be averaged. So there are several key points in ALECH. First, we only use the combined graph to bridge when those reads fail to bridge in their own sample. So in other words, we will borrow the junctions from other samples only if there's good reason to do it. Second, we will leverage the information from other samples to do graph decomposition when we have like unphased edges to address. So remember, in the graph decomposition, we mainly follow the phasing paths. But what if edges are not covered by phasing paths? Let's see this edge E3 here. So which edge should be connected to on the right side? So in this case, and only in this case, we will consider all the samples. So basically, we consider the coexistence of E3 and each of the edges on the right side. So if they coexist in many of the phasing, in many of the samples, or do not appear in many of the samples together, then we will connect E3 to it. And last, we will extract features from both individual graphs and combined graphs to hope the random forest model can give better confidence score. So in short, ALECH is quite careful in using the information from other samples because it acknowledges the differences between different samples or cells so as to not create many transcript artifacts. Let's see some results. So we compared ALECH with 4 meta-assemblers, Transmeta, PsyClass, Strain Type to merge, and Scallop2 combined with Taco. So this is the result on 2 single-cell RNA-seq data, one from SMART-SIG3 protocol with 100 cells, and this one is from SMART-SIG3 Express with more than 1,000 cells. And again, we draw the precision recall curve, and you can see the curve for ALECH is above the curves for other methods. So note that we train the random forest model using the annotation in chromosome 1, but we test and compare it on other chromosomes. So there will be no data leaking. So this is the results from applying on bulk RNA-seq data, and we can see that the conclusion is consistent. All right. So I mentioned a couple of times about genome annotation or transcriptomes. So they mean the annotation of known transcripts. So they are critical infrastructures for genomic and transcriptomic studies. For example, they serve as the reference for isoform level quantification. And also, genome annotation is closely related with transcript assembly. So on one hand, one of the main purpose of developing those assembly methods is to annotate genome, is to build a transcriptome using RNA-seq data. And on the other hand, so those annotations will also serving as ground truth to benchmark, to evaluate assembly methods. So interestingly, there are 2 main annotations that are widely used. One is RNA-seq, the other is Ensemble or gene code. Ensemble or gene code are quite similar to each other, but RNA-seq and Ensemble are quite different from each other. So RNA-seq in general is smaller, and Ensemble is larger, and Ensemble contains a bunch of computational predictions. So we studied the 2 annotations based on the striking observation we made, which I describe right now. So like again, the annotations can be used to evaluate assemblers. So we evaluate strain height 2 and scallop 2 on a bunch of data aligned by 2 aligners and onto 2 different genome builds. And we end up with like 68 instances. And here's the result. So when we evaluate them using RNA-seq, it's 38 to 30. So strain height 2 is better. But when we evaluate them using ensemble, it's 0 to 68. So scallop 2 is better. So this is quite interesting, right? Because opposite conclusions come up when we use different annotations to evaluate those assembly methods. So here's another observation. So we compare the differences of the accuracy when we switch annotation. So the arrows points from RNA-seq accuracy to ensemble accuracy. Like here, so when we switch the annotation from RNA-seq to ensemble, you can see the accuracy of scallop 2 increases in terms of both precision and recall. But you observe the opposite trend for strain height 2, right? When we switch from RNA-seq to ensemble, both precision and recall drops. So clearly, like scallop 2 likes ensemble and strain height 2 likes RNA-seq. So why is that? We investigated and we figured that out. So this shows the breakdown of ensemble annotation by their biotypes. So the largest one is protein coding transcripts, like there are 86,000 of them, and about half of them are annotated in RNA-seq. Second largest biotype is long non-coding RNA, and the third one is like returned intron or transcripts with intron retentions. And you can see the difference is huge. There are only 400 of them are annotated in RNA-seq. So in short, ensemble annotates a lot of transcripts with intron retentions, and RNA-seq only annotates a few. And then let's see the assemblers. So this is the breakdown of accuracies of scallop 2 and strain height 2 evaluated using ensemble. And this is the number of correct in terms of ensemble annotation of protein coding transcripts. This is intron retention. This is long non-coding RNA, etc. And you can see this one has the largest difference. And I think that explains this mystery. So scallop 2 predicts lots of transcripts with intron retentions, and those will be evaluated by ensemble as true positives, but as false positives by RNA-seq. And that's why we observe this like opposite conclusion. So in terms of methods, why scallop 2 assembles more transcripts with intron retentions than strain height 2? So again, this is because the nature of scallop series, it tries to preserve all the phasing paths. It tries to respect all the reads. So if there are reads that span exon and intron regions, so scallop 2 will assemble it, like leading to transcripts with intron retentions. On the other hand, strain height 2 uses a greedy strategy. Each time it finds a path from S to T and then assign reads. And this approach is not that sensitive to the reads in the intron regions. So we developed a tool called IR tool to identify transcripts with intron retentions in assembly. And we show that scallop 2 combined with this tool will give us a higher accuracy than strain height 2 evaluated even with RNA-seq. So in other words, if you don't like the transcripts with the intron retention, you can first run scallop 2 and then use this tool to get rid of them. And the result will be better than using strain height 2. All right, let me summarize. We started the theoretical formulation of transcript assembly, and then we developed those assemblers. Scallop is assembler for short reads RNA-seq data. Scallop RR is assembler for PacBio long reads. Scallop 2 is assembler for multi-end or paired-end RNA-seq data. Teros is assembler for circular RNAs, and Alech is for assembling multiple samples. And we also studied the difference between RNA-seq and assemble and developed this IR tool, which is to identify transcripts with internal retentions in the assembly. So is transcript assembly a solved problem? Absolutely no. Currently, we developed interest in using machine learning and AI techniques to do assembly. So we have used like a random forest model to score transcripts, but here we want to do like end-to-end assembly. And also we are developing methods to assemble certain types of data like nanopore data or ruling circle amplification data. And we are also very interested in the downstream use of assemblers for biological or biomedical discoveries. So we are happy to see that our tools are downloaded more than 30,000 times. So people are really using our tools. And here's one example. So Scallop was used to identify a novel ACE2 isoform. So ACE2 is the main entry point for SARS-CoV-2. And this new isoform was shown to be upregulated in response to interferons. I think with that, I conclude. I would thank both Kingsford Group at Carnegie Mellon and my group at Penn State. And the co-authors of the papers I introduced was highlighted here, Carl, Laura, Qiming, Qian, Tasvia, and Carl. And I want to thank Dr. Carl Kingsford again for his supervision and continued help. So, and of course, the funding agencies. And thank you for your attention and happy to take questions.
Thanks, Mingfu. So we have time for a couple questions, like 5 minutes worth of questions. I don't know if we have any online. If you have questions, either shout really loud or come to the microphone. It seems that the mics are picking up from pretty far away. So.
Yeah, fantastic talk. There is a lot of report to show that in our genome there's a lot of macroexon.
Yeah.
Macroexon. That is very difficult to be mapped,
Right.
and sometimes it will in the tumor tissue it will generate new antigen that may affect give us the target for the immunotherapy. So but in your reference-based annotation, is it possible to identify this kind of?
Yeah, I said it's very, very challenging. So the tiny exons are very small. So alignment is very hard. So therefore, we are considering using a hybrid assembly method. So those like tiny exons, we don't actually map them, right? We keep them like as unaligned. So like some part of the like reads that can be aligned accurately, we use that. Otherwise, we keep the read itself. Yeah.
I see.
So that could be one possible solution.
Thank you.